Good morning, dear students. In the last class, we just started the endoplasmic reticulum and have learned till its composition. Okay. Then sarcoplasmic reticulum. Sarcoplasmic reticulum. I already told you that sarcoplasmic reticulum is nothing other than smooth endoplasmic reticulum itself, and it is only associated with the muscle cell. So, what is sarcoplasmic reticulum is clear by this now to you. And now next we are going to see which are the components of this sarcoplasmic reticulum. Three components are present. Transverse component is always called a T system. A longitudinal component it surrounds the muscle cell, and a junctional complex. I will explain to you. Transverse component. Look in this picture. Transverse component or T tubule. Here we can see a green portion. That is the T tubule. And longitudinal means that part of the sarcoplasmic reticulum that runs longitudinally and cover the myofibrils. This portion, that is longitudinal longitudinal component. It surrounds the muscle cell. Here also we can see that this is the T tubule or transverse component. And both these components need to get connected each other. For that, junctional complex is present. In this picture is very clear. This is the T tubule. Okay, and this is the longitudinal component. Longitudinal component up to here. This portion. And this longitudinal and this T tubule need to get connected together by this junctional smooth endoplasmic reticulum or junctional complex. So that is the function of this junctional complex or junctional component of the sarcoplasmic reticulum to connect T tubule with the longitudinal component. Okay. Next. So I think it's clear to you. Transverse component, then longitudinal, and the junctional component. In this picture, it is not that much clear. Anyway, I think as I explained to you, now it's clear to you what is junctional complex also. Then functions of rough endoplasmic reticulum, to its function. For your examination, it can also as like an essay question about, write an essay about endoplasmic reticulum, explain its functions, like that way the question can come. So we are moving to the functions of rough endoplasmic reticulum. It helps in assist in protein synthesis. Okay, rough endoplasmic reticulum, I already told you that as ribosomes seen attached with that, it helps in protein synthesis. And also it involved in the processing of nascent polypeptide. Polypeptide is the first stage for forming a protein. So after forming a polypeptide, it needs some modifications by adding some groups, some sugars or anything like that. For that modification that is happening inside the rough endoplasmic reticulum. And protein glycosylation, that's what, the addition of sugar to the polypeptide. That is protein glycosylation also happening inside the rough endoplasmic reticulum. Then protein synthesis. Protein synthesis means functional difference of two types of ribosomes we need to understand. Ribosomes are two types exist in cytoplasm. Two types means those ribosomes, this is cell, those ribosomes seen free in the cell, and some ribosomes we already understood that is seen as it is seen attached with the endoplasmic reticulum. Okay, these are the two types of ribosome. But in what way both these ribosome functionally differ? Uh, I will explain to you. Free ribosomes, they link with the mRNA and it carrying out the translation while remaining free in the cytosol. Then coming to the membrane-bound ribosome, what they did, they synthesized the protein. We are explaining about this type of ribosome. They attach with the mRNA mRNA for translation. Translation means they need to read and understand and to decode the message written in mRNA to a polypeptide. That process need to happen here. Okay. And as these ribosomes are found attached with the endoplasmic reticulum, that polypeptide. That polypeptide, newly forming polypeptide is first entering in the cisternal space of the endoplasmic reticulum. Okay, and that means the lumen of endoplasmic reticulum. From that lumen, it is then transporting to the lumen of Golgi complex, because all these structures are interconnected. From its lumen, it can reach the lumen of Golgi complex, and from there it can reach the lumen of lysosome. Okay, that is the difference between the two types of ribosome. Free ribosomes and that ribosomes found attached with the membrane of endoplasmic reticulum. Okay, here it is directly discharging that polypeptide to the cytosol. Here it is not possible. That newly forming polypeptide is first released into the lumen of the endoplasmic reticulum. Okay. Next, we are going to see the processing of nascent polypeptide. In what way that processing the newly formed polypeptide, how it enters the lumen of endoplasmic reticulum, and what modification is happening to that we are going to see. In this picture, we can understand that. This is means this black line. This is mRNA. And I can see some ribosomes attached with this mRNA, and they are carrying out the translation. Translation means mRNA is converted to a polypeptide. That process, translation. So that ribosome is carrying out the translation, and that newly forming polypeptide we can see here. Its end portion we can see a special area. Here also we can see that sequence, that amino acid sequence is called a signal sequence. It is a signal. And to that signal, some protein came and bind. That protein are called signal recognition protein, SRP protein, signal recognition protein. They come and bind, and it acts as a driver or a director. It directs that ribosome along with that nascent polypeptide towards the endoplasmic reticulum, and in the membrane of the rough endoplasmic reticulum there present a receptor called SRP receptor. SRP receptor is otherwise called that signal recognition protein receptor. Receptor is present, and this complex means SRP protein along with the ribosome and along with the nascent polypeptide bind to that SRP receptor. Okay, until here I hope you understand the process. And after what will happen, SRP protein is released. SRP protein is released here, and then from that ribosome, that nascent polypeptide reach the lumen of the endoplasmic reticulum. And there present another enzyme called signal peptidase. Signal peptidase is present. Okay, and what is the role of this signal peptidase? The signal peptidase cleaved that signal sequence away from this nascent polypeptide. Okay, then that completed polypeptide chain, it is then released. Here we can see that it is released here for packaging and transport to other cellular organelles like Golgi complex and lysosomes. I hope it is very, very clear to you. Okay. Next, protein glycosylation. What is mean by protein glycosylation? Protein glycosylation means it's a process of adding sugar to the newly formed polypeptide chain. Here, we just only we understood and learned that newly formed polypeptide chain reach the lumen of the endoplasmic reticulum. After it reaches the lumen, some addition or modification or like addition of sugar units is happening inside here, and that process is called protein glycosylation. So that process is happening inside the endoplasmic reticulum, and that process is carrying out with the help of an enzyme called oligosaccharyl transferase. This is the enzyme that catalyzes the reaction of adding sugar to the newly formed polypeptide chain within the lumen of endoplasmic reticulum. Okay. Function of rough endoplasmic reticulum is over. Then moving to functions of smooth endoplasmic reticulum. Smooth endoplasmic reticulum, we already understand that it is concerned with the lipid synthesis and also the synthesis of steroid hormones. And also it helps in detoxification of poisonous materials, especially those materials that reach the hepatic cell or liver cells. And also it helps in intracellular transport. Intra means transport within the cell. Intra. Okay. Then protein glycosylation. Again, that process happens here also. Storage of calcium 2 plus ions. It stores calcium 2 plus ions, especially where inside the sarcoplasmic reticulum. Those smooth endoplasmic reticulum seen associated with the muscles. Inside that calcium is stored. And what is the role of this calcium ions? These calcium ions are very necessary for muscle contraction. How it helps muscle contraction, everything you will learn in another chapter. Now I am not going to explain that now. Okay. Then lipid synthesis. Smooth endoplasmic reticulum produce lipoprotein particles for the hepatic cell. That is, in that way we can tell that smooth endoplasmic reticulum involved in lipid synthesis. It produces lipoprotein particles. And also, I already told you that it involved in the synthesis of steroid hormones. In what way? It helps in synthesis of steroid hormones of which cells? Of endocrine cells of gonads and adrenal cortex. Gonad and adrenal both are endocrine glands, and its cells need to synthesize hormones, especially steroid hormones. For that, who is helping? Smooth endoplasmic reticulum itself is helping. Then detoxification to remove the toxic effect of certain materials like lipid soluble poisons, drugs, and many harmful compounds produced by metabolism. All needs to get detoxified, and that detoxification is also done by this smooth endoplasmic reticulum. Storage of calcium, I already explained to you. It is done by sarcoplasmic reticulum of muscle cells. Intracellular transport means it is the continuity of endoplasmic reticulum. I already told that endoplasmic reticulum is a continuous network from a nucleus till the plasma membrane. So, in that way, it can help for intracellular transport. So it facilitates transport of materials between cytosol and nucleus also. Uh, it provides increased surface area. We understand that its structure is a highly enfolded network-like structure. So that folded structure, branched network-like structure, increases the surface area. As its surface area is increased, that means it is getting that much space to accommodate key enzymes and their products. That is the significance. And membrane source means it provides a source, or it acts stand as a source of membrane for other membrane bound structures like a nucleus and Golgi complex. If Golgi complex or nucleus need to have some membrane parts, then they are depending this endoplasmic reticulum. Then role in gluconeogenesis. What is its role? Suppose this is a hepatic cell and this is the smooth endoplasmic reticulum. Okay. And we know that inside the liver cell, glucose is stored in the form of glycogen. And when the body or that cell require energy, the glucose need to release. That information is received in the form of a hormonal stimulation. Upon receiving that hormonal stimulation, that glycogen first converted to glucose 1-phosphate. Then glucose 1-phosphate again converted to glucose 6-phosphate. In all these steps, some enzymes playing important role that is not discussing here. Then glucose 6-phosphate with the help of glucose 6-phosphatase enzyme, it becomes free glucose. As it becomes free glucose, it can easily diffuse into the lumen of a smooth endoplasmic reticulum and can release outside the cell. And in that way, smooth endoplasmic reticulum helps in gluconeogenesis. Okay. Then biogenesis of endoplasmic reticulum. Biogenesis of endoplasmic reticulum, it is a multi-step process. What are the steps? Need to organize and organization of that first lipid bilayer, and need to synthesis its component protein parts. So that all are multi-step process. And also along with that its component enzymes, specific sugars, lipids also need to added. All these process is an elaborate multi-step process, and it takes place in a gradual pace. Okay, with this we finish this chapter and today's section. Thank you.